Yeah, hello, my name is Carsten Allefeld. I'm filling in for John Dylan Haynes, who can't be here this year because of some medical problems. And I'm going to talk to you about decoding and predicting intentions as a case study for MVPA applications. In a way, a case study for MVPA in cognitive neuroscience applications. Why do we think that we might be able to decode intentions from the human brain? The idea is that in the brain, if a person has a certain intention, there's a certain neurocorrelated, distributed activation pattern. And in fMRI, we can pick up some of that pattern in the form of the pattern of activation across a set of voxels. We use the set of this activation pattern across these voxels, form a pattern vector out of them. Then we obtain such data in several different situations where people hold different kinds of intentions. For instance, I have to feed my cat, I have to prepare a lecture, I have to book a flight, and so on. We train a classifier in order to associate these intentions with patterns, and then we test whether this classification works, whether there is actually an association between the patterns and the intentions by providing test data, independent test data, and hope that the right intention is being attributed by the classifier to this test data. Here's one experimental paradigm in which we have investigated this. This experiment was performed by Stefan Bode, who was previously a PhD student in our lab. He's now at the University of Melbourne. And this experimental trial consists of a cue, a delay, a stimulus, and a response phase. The cue gives the subject the information which task set or which kind of rules to follow in order to form intentions. Then there's a visual stimulus to which to apply a certain rule that is given to the subject. And then there's a response, which is a motor response, movement of the joystick to the right or to the left. Now, the task of the subject is, under the condition A, to associate one of the two color patterns, two color stimuli, with the movement to the left, the other one with the movement to the right, and under the other task set, the other instruction B, they are to reverse this association, use stimulus 1 in order to induce a movement to the right, and stimulus 2, a movement to the left. From this design, we can try to decode several different things. Of course, we can try to use a classifier in order to identify the target stimulus from the neuroimaging data. We have two different visual stimuli. We can try to see whether there are patterns in the brain corresponding to these target stimuli. We can try to decode the response. Is there a movement to the left or to the right of the joystick? But most interestingly, in this experiment, we can try to decode the mapping. In this given trial, has the subject been following the rule to associate stimulus 1 with a movement to the left and stimulus 2 with a movement to the right, or the other way around? So to try to decode the task set that has been indicated to the subject at the beginning of the trial using these cues A or B. Now, how to do this? In this experiment, there are 4 different child types or experimental conditions. And in order to access different kinds of information that might be in the data, we associate different kinds of classification labels or classifier labels with these 4 kinds of trials. So if we want to look at information about the stimulus, stimulus 1 or stimulus 2, then we put the same labels as label stimulus 1 to the 2 conditions on the left-hand side, and label stimulus 2 to the 2 conditions on the right-hand side. If we're interested in decoding response, movement to the left or to the right, we use this labeling scheme, response L, response R, to the 4 different experimental conditions. And finally, if we're interested in decoding the task set, we label the 2 upper experimental conditions as task set A and the 2 lower experimental conditions as task set B. But what do we put into the classifier in order to allow it to infer something about stimulus, motor response, or task set? Basically, there are 3 different approaches what we can do. The simplest is to just use all the voxels in the brain, feed it to the classifier, and hope that it can deal with this. This is not optimal. Many classifiers have problems with very large dimensional feature spaces and not as many data points. Also, it's not really biologically plausible to think that the information about a specific thing, a specific aspect of the cognitive experiment would be distributed across the whole brain. We can use a region of interest approach. This could be based on an anatomical atlas. We use all the voxels from a specific region that we picked beforehand, feed those data into the classifier. Or what we most of the time we do in our lab is to use searchlight decoding. We use a small group of voxels that have a shape like a small cluster around a center cluster. We do the MVPA analysis on this small group of voxels, which is typically something like 100 or 200 voxels. There's a result coming out of this analysis, usually something like a decoding accuracy. Then we associate this decoding accuracy with the center voxel. And this thing we do for every voxel in the brain. So every voxel in the brain is once the center voxel of a searchlight. So in the end, we get a map of local information content. Advantage of this approach is that we don't give it too much of a challenge to the classifier. We have a relatively small subspace. We use a locality assumption about the localization of cognitive functions in the brain. And we do not have to have any assumptions beforehand and where in the brain information about the specific thing we're looking at might be present. There's a dimension time. How do we select data temporally to provide to the classifier? Again, there are 3 different basic variants. We could use single volume activity. For each trial that we're looking at, we use one volume that is associated with this trial and use just the fMRI bold data from this trial in this specific volume. We could use a GLM approach. We could fit a general linear model to the data, but use a design matrix where we have one regressor, not per experimental condition, but one regressor per trial. So we have very many trials. We have very many parameter estimates, and each parameter estimate is associated with a given trial, and then we can do trial-level decoding, but based on a GLM. The thing most of the time we do in our lab is to use run-wise GLM parameter estimates of a regular model. So we use a design matrix as we would use it in a regular univariate analysis, one regressor per experimental condition. So we get one parameter estimate in each voxel for each experimental condition. But the experiment has several sessions or runs. There are several blocks of data recorded subsequently. For each run, we get separately parameter estimates. And then we give the classifier data from a subset of the runs and test it on the rest of the runs. So we do decoding on run-wise GLM parameter estimates. The practical implementation looks like this. At the beginning, we have the raw data, the raw fMRI data from several different runs. We apply the usual pre-processing steps like immersion correction, slice time correction, maybe normalization, reslicing, and so on. So we have a pre-processed data. Then we apply a standard, as we would use in univariate analyses, GLM analysis to these data. In this case, we have 4 different experimental conditions. So we have a model with 4 regressors of interest. So we get 4 different parameter estimate maps in each run. And then we train the classifier on data. In this case, we have 4 different runs. We train the classifier on 3 of these runs, test it on the left-out run. We do this for each searchlight center. And we do this for these 3 different labelings of the experimental conditions corresponding to the different kinds of information we are interested in. We do this, in a circle. So each run is once used for test data. The other 3 runs are used to train the classifier. So we have something what you might call leave one run of cross-validation. And the result of this is for each single subject that we have a map of local decoding accuracy, for instance, or some other multivariate pattern analysis method. In this case, it would be a decoding accuracy map. Of course, one for each of the 3 labelings of the experiment conditions, so for the 3 different analyses we are interested in. The first 3 steps can be done in a standards package for fMRI data processing, for instance, in SPM. Now, as a result of this, we'll have for each single subject an accuracy map. What we do is this on the second level, we do the same thing as we do in univariate analysis. We average these decoding accuracy maps. Most of the time, we also smooth them in order to further reduce problems of realignment, spatial normalization between, so differences in anatomy between different subjects. So we have an average smooth map of local decoding accuracy. And then we can apply a t-test across subjects, for instance, or if we don't want to use a t-test, for instance, some permutation-based procedure, and we get a thresholded map of local decoding accuracy. And then interpretation would be, because this is based on a searchlight approach, that these blobs that we find in there stand for local presence of information about the kind of labeling that we put into the classifier, that we use in order to train and test the classifier. And again, this last step, after the decoding, the single subject decoding accuracy maps have been computed, you can do in a standard package like SPM if you're okay with a t-test. Now, for this is the experiment. We can do stimulus decoding, with stimulus 1 versus stimulus 2. Then, as to be expected, we get information about the stimulus, significant amount decoding accuracy, above chance in right and left visual cortex. I also put a bar chart here, which shows extracted decoding accuracies from 3 different points in the brain, one area in left visual cortex, one in right visual cortex, and one just for control purposes, doesn't make sense, from corpus callosum. The bars show the average decoding accuracy across subjects in this specific area, and the error bar shows the standard error of the mean. And what we find is that, yes, there is significant information above chance, significant decoding accuracy above chance, which in this case is 50% in both left and right visual cortex, but of course there's none in corpus callosum. There's some caution that has to be applied in interpreting these kinds of extracted data, because we look here at data that have been specifically selected from areas where there's a specifically large effect. These are not unbiased estimates, so we can't use them, we can't interpret them as estimators of the true effect size, of the true decoding accuracy. If you're more interested in this topic, you should have a look at the publication of Wu et al., who discusses this in a wider context. We can do response decoding. Then, as to be expected, we get effects, information in the fMRI data about left or right movement of the joystick in motor cortex, with also some parietal areas. And we can apply this to the task set decoding. Wow is the association between visual stimuli and according responses, and we get widespread responses, widespread areas in left frontal and parietal cortex, which contain information about this. The results I've shown you before are based on a standard canonical Hamilton response function, GLM. This is the way we extracted the parameter estimate that was put into the classifier. If you also want to know something about the temporal dimension, we can use instead a finite impulse response model, or FIR model. The difference between the two is that in the canonical HRF, for each experimental event, for each response, we have one model which has a certain shape, and what is being estimated is just the height of this shape, the strength of this response. So we have one parameter that's being estimated. In an FIR model, we have one parameter for each latency from the beginning, from the event itself. So we have one parameter for the onset, one parameter for onset plus 1 TR, one parameter for onset plus 2 TRs, and so on. This is how it looks like. As I said, in the canonical HRF, we have just one shape, one regressive shape which is associated with a parameter. In an FIR model, we have a set of regressors which have the same shape. It's basically a delta function or a stick function. There's a response at a certain time point and at no other time point. But we have a set of these regressors, one for each latency, one for the beginning of the trial, one for the next time point in the trial, one for the next time point in the trial. As a result, and of course, these regressors, these responses are repeated for all the experimental events in the corresponding experimental condition. And as a result, we get this typical diagonal line pattern in the design matrix. Now, we have parameter estimates for different latencies within the trial. And so we can apply a classifier, train a classifier, test a classifier, compute accuracies for each different latency within the trial. And so we have something like a time-resolved decoding analysis. This is how it looks like for the task set experiment. It's 3 different decoding analyses corresponding to 3 different kinds of labelings of the experimental condition. Across 5 of, I think, 8 in total FIR bins. In the decoding of the target stimulus and the motor response, nothing special happens, but there's something interesting if you look at the decoding of the task rules. In the first FIR bin, you see information about the task rule, which in this case is, at this point in time, is really a visual stimulus on the screen, the letter A or B, which indicates a task set to the subject. You get information in visual cortex. But this information moves further into further frontal areas over time, and this could be interpreted in such a way that the purely visual information from when the cue that indicates this information is being processed is transformed in a more abstract representation of the task set that is to be performed, that is to be executed. It's a second experiment about task sets. In this case, we looked at, in this experiment, which was performed by Carlo Verberi. He used to be a postdoc in our lab. He's now at the University of Milano. we were interested in the relation between simple and complex rules. In this case, a simple rule is just the association of a given visual stimulus with a given motor response. For instance, if you see a face, move the joystick to the left. If you see a house, move the joystick to the right. We have 4 different of these rules. And these rules are indicated to the subject, in this case not by letters, but by more abstract squiggles, which are learned by the subjects before the actual fMRI experiment, which are those symbols in the first column. But then we also have 2 complex rules, which just consist of the combination of each of 2 simple rules each. So the first and the second simple rule together make up 1 complex rule with its own associated squiggle, and the third and the fourth simple rule make up 1 more complex rule indicated by another squiggle. So in each trial of this experiment, the first symbol, the Q, can be 1 out of 6 different symbols, which instruct the subject to either apply a simple rule or a complex rule. Now, what we're interested in here is a pattern in the fMRI that represents a simple rule related to the pattern that indicates the presence of a complex rule. So if you have a complex rule, and this complex rule is just a combination of 2 simple rules, is the pattern that represents a complex rule the combination, the addition of the 2 simple rules, or is it a different pattern altogether? And this we can investigate using cross-classification. We train a classifier on some part of the data, we test it on some other part of the data with different labels. In particular, if we just decode the rule itself, we train a classifier to recognize one of the simple rules, and when we apply the classifier in order to find out what is the simple rule, then we just decode the rule itself, and we find information in a right frontal area and in a left parietal area. But if we try to decode the complex rule using a classifier that was trained on simple rules, we can do so in the right frontal area, but no longer in the left parietal area. So this indicates that there are different compositionality properties of the representation of simple and complex rules in different areas in the brain. I think I'm going to skip over some slides because I don't have enough time for that. But there are some things I would like to show you. There's a number of different pitfalls in applying MBPA analysis to fMRI data. There are, of course, pitfalls also in univariate analysis, but maybe more so, or additional pitfalls in multivariate analysis. In this case, we're looking at something that is common to multi and univariate analysis, the problem of overfitting. If we have a given experiment with a given set of experimental conditions or a given set of experimental events, we also always have options to differently model these experimental events, for instance, in the design matrix. So we can do different analyses. We can, based on these, for instance, design matrices, we have different parameter estimates. We can run different decoding analyses, and then in the end, we have to pick one analysis. And there's, of course, the possibility that one would pick the analysis where something comes out of it which you like or what you have expected. This is the problem of overfitting in neuroimaging. And here's a proposal to fix this problem, or at least partially fix this problem, using Bayesian model selection applied to general linear models. In this case, we have visual stimuli, which are small bars extended throughout the visual field. And there's one bar on the left and one bar on the left, which has a slightly different orientation. And we have 4 different orientations on the left, 4 different orientations on the right, so we have 16 different conditions in total. Simplest thing we can do is to have a design matrix with 16 different regressors for these 16 different experimental conditions. Another thing we can do is to just have one regressor that describes the onsets of these stimuli, and then 2 parametric regressors which describe parametrically the deviation, the angle, the rotation angle of this one bar on the left and another one on the right. So we have 3 regressors in total. And this can be done in a way that is linearly related to the angle or in a way that is nonlinear related to the angle. This method for Bayesian model selection applied to GLMs has been developed by Yoram Zoch, a PhD student in our lab. And here's the results that he achieved by looking at this data set and the different ways, possibilities to model it, these 3 different models, GLM1, GLM2, and GLM3. And what he found is that in most areas of the brain, there's a strong reason to prefer GLM3. According to Bayesian model selection, this is the way to model the events in this experiment. This is particularly true of those areas which we expect to be interesting in this experiment, which is primary visual areas and V4. If you're more interested in this methodology, please go to Yoram's poster. He will present it tomorrow, 12:45, and it's poster 2190, solving the problem of overfitting in neuroimaging, ? I would also briefly talk to you about an alternative to decoding that we can apply in studies with designed experiments in cognitive neuroscience called cross-validated maneuver. The idea here is that if we train and test a classifier, and we are not really interested in the single predictions, the single classifications, but we just summarize the results of the classification using a measure like classification accuracy, then this classification accuracy is actually a measure for multivariate effect strengths. And there are different ways to measure multivariate effects. One that is well known as the so-called Mahalanobis distance. In this case, instead of classifying the models, the distributions of data points belonging to different classes by multivariate normal distributions, and then we look at the distance in a voxel activation space between the centers of these distributions and measure this distance in terms of standard deviations of the distributions. This method has, for instance, been tested by Nicholas Krieger's-Carter. But this can be done more generally. If you have more than two classes, then we can look, for instance, at the variance spanned by the three condition centers, and then we compare the multivariate variance that accounts for the difference between the classes and compare it to the multivariate variance that accounts for the variation within the classes, which we, in this case, treat as error variance. So what we're trying to do here is to estimate the multivariate variance explained by the effect versus the error. And to do so, we most conveniently use the multivariate version of the general Neo model, which has exactly the same shape as the known univariate GLM, but with the difference that we look at multiple voxels at the same time. On top of this, this framework, which is well known, which is old in statistics, called multivariate analysis of variance, or MANOVA, we put cross-validation. And the nice thing about cross-validation here is that we can obtain an unbiased estimate of our effect strength, and we call this unbiased estimate of the effect strength pattern distinctness D. This method is particularly adequate if you have complex factorial designs, because we can now look for different kinds of effects, different kinds of information, not by assigning labels to experimental conditions, but by using contrast vectors in the same way as we do this in univariate analyses. And we can use this with multiple classes at once, with parametric regressors, with interactions, and so on. So it's a very versatile tool for MVPA and factorial analysis. Here's a short overview of some studies that we have done using cross-validated maneuver applied to visual working memory. These studies have been run by Thomas Christoffel, a postdoc in our lab, and Chang Yang, his PhD student. I'm not going into the details of the experiments, but in any case, there's some visual stimulus at the beginning of the trial, there's some delay period, and then there's a task where the subject needs some of the information from the visual stimulus. So we can assume that in the interim, there will be some kind of visual working memory storage of the pattern or of some information about the stimulus. Something that they found, Thomas Christoffel and Chang Yang, is that in this case, in different studies with similar designs in this area of visual working memory, it appears to be that the variance across subjects, the variation across subjects, as assessed by the pattern distinctness D, is more comparable between studies than this is the case for decoding accuracy, for instance. And this might lead us to believe that we can use this measure in order to make inter-study comparisons. For instance, in the Chinese character study, we find information in Broca's area. We do not find information in Broca's area for the color patterns or for moving dot patterns. But now we can not only say there's significant information here, not significant information here, but we can also say there's a significant difference between, for instance, study 1 and study 2 as regarding Broca's area. If you're more interested in these experiments, please go to OHBM poster 2177 by Chang Yan, presented on Tuesday with the name Decoding Neural Representations of Chinese Characters Stored in Working Memory. To finish the talk, I would like to briefly tell you about an experiment where it's not just possible to decode present intentions, but also to predict decisions, which is a kind of predicting intentions that have not yet been formed, actually, at least not consciously. The study has been performed by Chun Seong Soon, a former PhD student in our lab, who is now at Duke University in Singapore. In this experiment, on the screen there was a sequence of random letters presented to the subject, here B, K, Y, P, Q, and so on, one at a time, changing every 500 milliseconds. And the subject was told that he or she could freely decide at which time point to press a button and freely decide whether to press the left or the right button. The subject was also asked to memorize the character that was on screen at the time where the subject felt that he was consciously aware of the decision. He knew he was going to press a button, and he knew he was going to press this specific button. Now, by this indication of the subject later, we can estimate the time point at which the decision to press left or right became conscious. And then we can try to predict from the fMRI data the left or right button press, either from data before the time point of conscious awareness or from data after the time point of conscious awareness. If we look at data after the time of conscious awareness, we find information about the upcoming button press in left and right motor cortex, in the supplementary motor area, in pre-SMA. But if we look at data before the time of conscious awareness, we find information about the upcoming button press, left or right, in lateral frontopolar cortex, medial frontopolar cortex, precuneus, or posterior cingulate cortex. There is one possible, or actually there are probably several possible criticisms of the study, but one possible criticism of the study is that there are strong sequential dependencies in the behavioral data. People do not press randomly left or right in subsequent trials, but there are strong dependencies. If they press the left button in one trial, there's a probability of about 65% that they will press the right button in the next trial, or there will probability of 35% to stay with the current choice. This might suggest that what we're seeing in the fMRI data is not really a decoding of the next button press, but of the memory signal, of the representation of the previous signal, which is correlated with the next button press. So we're not really getting the kind of information we're interested in. We checked this hypothesis by explicitly trying to decode not the next button press, but the previous button press from the data. And we found that in those areas where we are able to predict the next button press, we are not able to predict the previous button press. So at least on this level, it appears that we're really looking at information that is related to the forming of an upcoming decision and not to the previous decision, which is correlated to it. A quick summary. MBPA and decoding can be used to investigate the neurorepresentation of intentions, even of decisions before they become conscious. And I've gone over a number of important techniques that to use in this context. Something that is general to do MBPA, I think, is training and testing of classifiers to assess information. Interesting maybe also that we can use different associations of classification labels with experimental conditions in order to assess different kinds of information. We can use the searchlight in order to have a localized assessment of information. We can use the leave one run out cross validation on run-wise estimated GLM parameters for classification. An advantage of this is that we have well-defined pattern estimates. We can use FIR modeling instead of HRF modeling for time-resolved analysis. And we can use cross-classification, for instance, to examine commonality or invariance of representations that are related. I didn't have that much time to talk about the pitfalls, but I think I presented to you the idea of avoiding overfitting, for instance, by Bayesian model selection of GLMs. And I also briefly talked about cross-validated MANOVA as an alternative for decoding and a natural framework for MBPA in the context of complex sectorial designs. Thank you very much. Are there any questions?
Everyone is a bit hard.